Hi everyone, Kevin back here with video number 4 of 4 in cytoskeleton and intracellular transport. This video is just an hour long. Just kidding. This video is short. We're going to finish off muscle contraction. We left off here with the cross-bridge cycle. And we said that when ATP binds and then ATP is hydrolyzed, we get this change in conformation in myosin, and that contacts actin, and it moves the thin filament toward the center of the sarcomere. So what if ATP is around? Is this just automatically going to happen? Well, no, of course, we control our muscle contraction. So how do we control it? That's the last step of the puzzle. You saw some of that previewed, or actually pretty well explained, in the video at the end of that I referred you to at the end of video number 3. That description was of the process of calcium triggering muscle contraction. So in video 3 we talked about how the actin filaments, microfilaments, are surrounded by this tropomyosin, all right? And that's the long fiber here that blocks the sites. So the little indentations here in the actin monomers, that's where myosin's going to bind. And those sites are blocked. Myosin binding sites are blocked by tropomyosin. What we didn't see in video 3 in that picture was the troponin complex that's part of the tropomyosin binding or blocking of the binding sites. So this tropomyosin wraps around actin filaments, blocking the myosin binding sites. So that's sort of the normal state of affairs. Well, how do we change that so myosin can bind and our muscles can contract? Calcium changes that. So calcium ions will bind the troponin, and that'll change the conformation. Same story, binding, change conformation. That changes the conformation so tropomyosin no longer covers the binding sites. So calcium is our secondary messenger of sorts that takes the signal from our neurons and translates that into muscle contraction. Now, there's a couple steps before calcium actually does its thing. Let's look at those. First, finishing off this slide, a couple more words here. So troponin binds calcium and shifts the tropomyosin out of the way. All right, that's the story on this slide. So those other steps that get from our brain thinking muscle contraction to it actually happening. It starts with a neurotransmitter opening a ligand-gated ion channel. So what does that mean? All right, so here is our nerve, and our brain says, make my muscle contract. And the nerve then sends acetylcholine, the messenger, down to the synapse, right? So here's the synapse, that junction between the neuron here at the top in yellow and the muscle cell at the bottom. So acetylcholine crosses that synapse and binds to ligand-gated ion channels shown here in the green embedded in the cell membrane of our muscle. So we know a little bit about signaling and signal transduction now. So you recognize those ligand-gated ion channels. Make sure you know those well. The ligand that binds is acetylcholine. It's the ligand that opens the ion channel. The ion that flows through in this case is sodium, the little purple circles here, spheres shown in our illustration. So acetylcholine binds the ion channel, causes it to open, and then sodium flows through into the muscle. Okay? So, muscle fires, acetylcholine comes down, it binds the ion channel, sodium enters the muscle cell. So I said there were a couple steps before we get to calcium. Let's look at calcium then. Oh, here's a little more. That's what we just saw. So here's a summary. Acetylcholine binding opens the channels. Sodium flows through. Sodium changes the membrane potential of the muscle cell. not the neuron, but the muscle cell, we've got positive ions flowing in, so it changes the potential across that membrane. And he talked about that a little bit in the video at the end of our video 3. It's that change in potential that causes the calcium release from the endoplasmic reticulum, and that starts muscle contraction. So there's how we get the calcium. The sodium triggers the release of calcium from the endoplasmic reticulum, and actually we call that the sarcoplasmic reticulum in the muscles. It's a specialized version of the endoplasmic reticulum which stores calcium. So here's the full picture. So there's the neuron up at the top, releasing the acetylcholine. Sodium flows through. That's kind of step 2 here. Sodium flows, and it enters this it triggers the release of calcium from the sarcoplasmic reticulum. So these are these channels down here, where I'm drawing the arrows now. Those are the channels in the sarcoplasmic reticulum which release calcium into the cytosol. The calcium then, of course, binds to the troponin, releasing the tropomyosin, allowing the muscles to contract. Okay, so step 1, firing of a neuron. Step 2, sodium flowing. Step 3, the change in the membrane potential triggers the release of calcium, opening the ion channel in the sarcoplasmic reticulum. Remember, we said when we talked about ion channels, and we didn't talk about it much, but we talked about how membrane potential can actually trigger an opening of an ion channel. Here's an example of that. Calcium comes out, binds the troponin, tropomyosin clears out the myosin binding sites, myosin binds, and we have muscle contraction. And then we start over again as the process cycles. Now, the last thing is there's a calcium pump that reuptakes the calcium back into the sarcoplasmic reticulum. Because if we had calcium present and we had ATP present, our muscles couldn't relax. All right? So when our neuron stops sending the signal for our muscles to contract, the calcium stops flowing out of the sarcoplasmic reticulum. So that process stops. The calcium pump takes calcium back up, and our muscles relax. That's it. That's muscle contraction. Thanks for watching. Know this pretty well for the final because it will definitely be on there. And I will see you in class.